More and more these days, modern medicines are manufactured by bioprocessing rather than just wet chemistry. So aspirin, for example, is just a chemical formula that's pretty easy to manufacture. But modern biologics are complex proteins, and these are manufactured using live organisms, bacteria or sometimes animal cells. And what the Prima does is just monitor how well those cells are thriving in the medium. And if things go a little bit wrong, then we can monitor that with a mass spectrometer and correct what is a very expensive manufacturing process. The basics of a fermentation process are a bioreactor or fermenter, which can be single use or it can be a stainless steel reactor that is sterilized. And then we add a growth medium, which is chemically defined. So that is designed to support the growth of a particular microorganism. That could be a bacteria or it could be a yeast, insect cells, even plant cells, and animal cells. And those cells are typically modified in order to produce a particular target molecule. So where we use the Prima is in developing that process from the very small scale, starting at 2 liters, all the way up to an industrial scale process, which can be hundreds of thousands of liters. So the analytics that we provide are used in that scale-up process to get the product to market much more quickly than you would if you didn't have online analytics. At BPI, we're concentrating on bioprocesses, so that would be active pharmaceutical ingredients, obviously therapeutic proteins, but also vaccines. And then outside of the pharmaceutical space, there's fuel cells, there's industrial enzymes, and then we also do research in the chemical lab. We've had some very good testimonials from people who just enjoy the precision. There are lower cost mass spectrometers based on quadrupole technology, but the precision isn't there to get very precise respiration metrics. So the fact that we've now got a magnetic sector in that same benchtop form factor means they can use that high precision to really tease out the complexities of the manufacturing process much sooner and get to scale up much more quickly. So really it's a plug and play analyzer, very simple calibration, but calibrations are probably months and months apart. So the stability, the precision, really mean it's a very low maintenance analyzer. Well, the first thing about mass spectrometry is we're making our measurements after the bioreactor. So we're simply making gaseous measurements in the effluent. So we're not risking in any way the contamination of the batch. And it's a very information-rich measurement, so we can compare the gases that we use to sparge the bioreactor or the fermenter and compare that with the gases in the effluent and make very precise measurements of oxygen consumption and carbon dioxide evolution, so that we're calculating these respiration metrics, which give us a very good picture of exactly how healthy the cells are, and we can measure very precisely when things start to go wrong, long before you can detect those deviations with any other technology.